Good afternoon, everyone, and thank you for joining today's webinar where we will feature Marvel Biosciences, listed on the TSX Venture under the ticker MRVL. I am joined today by my colleague Babak Pedram, and of course, Marvel President and Chief Scientific Officer, Mark Williams. At the end of the presentation, we will take time for Q&A. Please use the Zoom chat function to submit your questions, and we will be sure to get to them at the end of the presentation. On that note, I will hand it over to you, Mark, to begin the presentation. Mark, please go ahead.
Thanks very much, Corey and Babak, and thanks to everyone for their time today to listen to the presentation. Here's our standard disclaimer. I plead the fifth in this case. You can read at your leisure. In a nutshell, Marvel is a biotechnology company focused on the development of new derivatives of known safe drugs. And by doing that, we call that drug redevelopment. We believe that we significantly de-risk the time, the cost, and of course, the risk in developing a new molecule with no known previous toxicology or clinical background for clinical trials. And so we think that this is a much more de-risked approach to traditional pharmaceutical development. And not only that, but we're also developing the same asset for potentially multiple very different diseases, so that if, for instance, the molecule is not active in one sector or one disease, such as in the CNS, there's a very good chance that it might be active in a non-CNS related disease. The company has got about 50 years of experience in either capital markets and/or biotech compound development. We have multiple assets. I'll talk about two today specifically. There's three major disease areas with an aggregate market value of in excess of $250 billion. Our approach, as I mentioned, is this idea of redeveloping drugs, and there's some very good historical examples of that. One of which is Celgene, which took a compound called thalidomide, which you may know from its kind of checkered past, as starting off as an anti-morning sickness nausea agent, and it was later discovered to have some fairly potent anti-cancer activity. And Celgene redeveloped it, that is, it tweaked certain elements of the molecule and developed a new, less toxic molecule, as it turns out, called Revlimid. That's certainly a major seller for them, and they're part of their franchise going forward. So that's an example of a large biotech, successful one doing it. The second example is Aurinia, which is actually a Canadian story, which started off taking cyclosporin, which is used to treat organ rejection. That was modified to make voclosporin, and voclosporin actually got FDA approval, and there was actually a big transaction for Aurinia recently that was developed for lupus nephritis. And so sometimes making these fairly small chemical changes can have introduced composition of matter, intellectual property. You have a very sense of what the toxicology looks like. You can usually improve that, or improve convenience, or improve the efficacy of a drug going forward. So this is a strategy that's been employed before very successfully to find new drugs based on old drugs for new diseases. So our lead asset is called MB204, and it's essentially a redeveloped derivative of a known drug called istradefylline. And we're looking at it for two predominant areas right now: depression and Alzheimer's disease, and also fatty liver disease, which we announced some data recently on. So istradefylline was originally developed as an anti-Parkinson's type of drug, and it's known to interact with the dopamine D2 receptor, and was approved by the FDA, I think in 2019. It turns out that the adenosine A2A target is actually a very interesting target and has applications to other diseases outside of Parkinson's. So specifically, there's good evidence that the target is involved in depression and anxiety. Again, there's good evidence that this molecule is involved in Alzheimer's, and there's actually even more papers coming out really highlighting the possibility of the adenosine A2A receptor as a target for Alzheimer's. And thirdly, it's a known target for fibrosis. In this case, we're looking specifically at liver fibrosis. So as I mentioned before, as a company having multiple drugs, multiple targets to go after, we think we can significantly de-risk this asset. Usually it's one drug, one target type of approach, but we have one drug, multiple targets that we believe we can pursue with 204. So here's some very early or initial data that we have. We've made new derivatives, fluorinated derivatives of istradefylline. We'll start being in a position to disclosing the actual structure very shortly. Filed composition of matter patents in March of last year. We're showing here that our drug has got fairly comparable activity against hitting the target in vitro, but by modifying the drug the way we did, which was a logical change with these fluorines, we were actually able to improve. On the right-hand side, you can see what's called the oral pharmacokinetics. That is, we're able to increase the amount of drug being absorbed and seeing it last in the body longer than the parental istradefylline molecule. So it appears to be significantly a very similar effectiveness in vitro, and looks like it has a slightly better PK in vivo. So to talk about the first sort of indication we're going after, the Alzheimer's and depression market, and there's a lot of overlap between Alzheimer's and depression, and in fact, depression is a very significant comorbidity factor with patients with Alzheimer's disease. And it's at least a $30 billion market. I think that's a fairly conservative number for this. And as you know, with the whole pandemic, the backdrop of the pandemic, there's been a significant increase in depression and anxiety for patients. The sales of prescription drugs for antidepressants have gone up, I think, as much as 30 to 50%. And even as many as 1 in 2 people are now sort of suggesting that they have symptoms of anxiety or depression because of the pandemic. And again, the major problem is it seems to be an issue with certainly the younger generation going forward, which is obviously a concern if this is weighing heavily on them right now. And of that type of treatment, the serotonin reuptake inhibitors are the most widely prescribed forms of those drugs, such as Prozac. But there's multiple issues. Some people don't respond to them. That's about 20%. It can often take 2 to 4 weeks for the effects to kick in. And in the case of certainly some of the serotonin reuptake inhibitors, up to about 40% of patients report sort of a loss of motivation and an apathy. In that case, they kind of lose their drive and ambition, which is a bit of a negative side effect for that type of drug. It'd be very good if you could come up with a drug that, had some antidepressant activity and actually could potentially motivate you, make you want to do more, get up and be active and so forth. And this might be the case with, again, going after the same pharmacology associated with the adenosine A2A receptor as well. So this is sort of indirectly and modulating the dopamine pathway. It's not an overt effect. It's almost like a tickling effect almost. This could be a potential first-in-class treatment for anxiety. And the way it works is by modulating the dopamine pathway, you're modulating the reward and motivation pathway, unlike the serotonin pathway, which is very, very different. What's also interesting is that we've got some pretty good evidence in our hands, and it makes sense that the dosage that you require to have an antidepressant effect is likely lower than the dose that you would need to, say, treat Parkinson's disease. So there's a potentially healthier use of the drug that way. And there's some evidence certainly been published that istradefylline has some clinical benefit in patients already in Parkinson's people with, say, depression or mood disorders. So we, again, as I mentioned, follow a drug redevelopment sort of pathway, and that allows us to say we feel very confident that the original molecule istradefylline is considered quite safe. And is certainly effective enough to be approved as a treatment for Parkinson's disease. So we're starting off with a molecule that's gone all the way through approval process already. And again, we're making some fairly logical but modest changes, logical and reasonable changes to the original molecule. And it certainly seems that there's no overt toxicological signal here with ALTs and ASTs when we dose head-to-head against istradefylline. Those are markers of liver health, rather. And again, we don't really see any effect on kidney health as measured by creatinine, but in this case, we see a very pronounced antidepressive effect in preclinical models of depression. And you can see here on the right-hand side in this forced swim test, a very prototypic model of depression. We see a very nice dose-dependent reduction in the activity of the animals And the nice thing is this is after 1 hour of dosing. This is not requiring 2 to 4 weeks of dosing to see the effect. So it looks like there's a fairly immediate effect with this drug, which is good. And again, very nice statistical significance. Those p-values, those stars there, reflect how strongly the statistics are showing you the effect is. So the more stars you have, the more effective it is. And typically in biology, 1 star, which is a p-value of 0.05, is considered significant. So anything lower than that is considered very good. So a second area that we're looking at developing this molecule for, again, to de-risk the asset, is in non-alcoholic steatohepatitis, NASH, is the acronym, or fatty liver disease. And this is estimated anywhere between a $20 to $80 billion market, depending on the source that you read. And it turns out that really the biggest issue with NASH and fatty liver disease is once you start developing fibrosis. And it's fibrosis that's the real problem. It sets you at a much higher risk for cirrhosis, liver cancer, which would ultimately require liver transplants. And you can see here, this is US figures on the left-hand side. You know, there's about 328 million, give or take, million people in the US with normal livers. There's about 67 million people with the start where they have a fatty liver. And then of that group, you can see a progression towards more and more fibrosis. And roughly 20% of the population may have steatohepatitis, steatosis, and NFALD. But you can live fairly comfortably with that. It's when it starts to develop the fibrosis that really appears to be the major issue. And unfortunately, right now, we don't have a lot of compounds in development. And I should say that there is no approved therapy right now, as far as I'm aware, for the treatment of NASH. There's been several that have been tried. And even within the massive class of molecules that go after NASH, the few that are really targeting the anti-fibrotic aspect of the disease, there's even fewer than that. So on this within the green arrows that you see there, we're highlighting the compounds that are targeting more of the fibrotic aspect of the disease. And if you pin down towards the middle, right near the bullseye, is where you can see sinacrivaroc, which was a repositioned drug that started off its life as an anti-HIV drug that found uses potentially treating fibrotic-related disease. Sinacrivaroc is currently in phase 3. It's the lead asset and represents only a small fraction of compounds in the NASH space that are actively going after the fibrotic-related disease. So it's the lead asset going forward. And we're basing our approach on what's already been published out there, and there's some very good evidence that blocking the receptor, specifically at hepatic stellate cells, which are responsible for producing collagen, and collagen is the material that makes up a fibrous clot or scarring that you see. Again, if you cut yourself, the collagen there is what gets deposited to try and tidy up and cause the scarring that you see there. And it looks like going after the adenosine A2A receptor, which is the target of our compound MB204, is related to the fibrosis process at the liver. And so we conducted 2 experiments with the compound in 2 different models. In the first study, we went and used a model from a group in Japan that's got an excellent model called the SMC Stellic model, where they were able to induce steatosis. And the score here is called a NAS score, which is a composite of several endpoints, and it's used clinically to see how patients are doing. It involves steatosis, ballooning, and inflammation. And so we see when we treat animals with our compound, our lead MB204, we're seeing a 1.4 reduction, which is reasonable, modest reduction in the NAS score. But the real one is on the second-hand side, the second study, where we went and studied, I think, 30 to 33-week-old animals who had already developed the NASH. These were pre-aged animals. And we're showing you here head-to-head against sinacrivaroc, that lead phase 3 asset. We actually saw a surprisingly very good response with MB204, about a 47% reduction in fibrosis compared to control. But actually, we outperformed sinacrivaroc, which which was a bit of a surprise, pleasant surprise to us. But again, we feel that this is a very reasonable comparison to go head to head. So we see that this is a de-risking of our assets. So we have CNS related diseases, and it looks like we can potentially treat or have an option to treat fatty liver disease related fibrosis in this case. So potential near value creation events. So in the Q4 of this year, we're looking to complete the preclinical testing of MB204. We just actually hit that milestone recently with the press releasing of our data on NASH. We're also pursuing a secondary indication with oncology. We are manufacturing our drug right now under CGMP conditions, and the first run multi-kilogram batch comes off this quarter. We're intending to begin our maximum tolerated dosing studies. That's what MTD is short for. This allows us to now confirm the safety of the molecule prior to giving it to humans in clinical trials. And we're finishing up a few more analogs of MB204, fluorinated analogs of the compound, so we can patent a bunch of molecules around our molecule, so to block people. And at that point, we would like to start reaching out to pharmaceutical companies to start gauging their interest. Those typically take time, so we'd like to start those discussions sooner or later. Q1 of next year, we intend to complete the CGMP run. So this is the material that will ultimately wind up into people. And we start beginning our 4-week GLP toxicology studies for our compound. In Q3, we intend to complete those toxicology studies and start having our file for our FDA pre-IND meeting. And by Q4 of next year, our hope is to get our phase 1/2 clinical trial underway with MB204. And interestingly, because I showed you earlier on, I mean, we can dose our drug, which is basically based off caffeine, by the way. And within an hour, we see measurable effects in animals. It's essentially the same thing with people. With caffeine, you take it in the morning, and you take it in the morning because you want to feel more invigorated later in the morning. So it has a fairly quick onset of effect. And we think that we can measure the benefit of our MB204 fairly quickly and potentially get an efficacy measurement in early Q1 of 2023. I believe at that point, we have an excellent chance of seeing a significant value creation event. I think it'll be another event to trigger interest in pharma and ultimately may help us in uplisting into US exchanges. I do want to touch on very briefly, we initiated fairly recently a second program redeveloping psychedelics, the treatment of depression and addiction. As you know, psychedelics got huge promise with treating all sorts of different diseases of the brain, particularly depression and addiction. But there's certain liabilities with certain compounds right now. In the case of ketamine, very good compound for depression. There's an abuse potential. And in the case of things like LSD, some of the tryptamines, there's a possibility of hallucinations. And they're very often given, because of that, given under medical supervision. So similar, we'd like to ask the question, well, what if there was a neuroplastic agent that you could take that didn't have these liabilities that you could take safely at home? And we think that this is going to be the next iteration of the psychedelic space. And I wanted to point out to you an example. A company called Delix Therapeutics recently closed $70 million in Series A financing. They're also developing molecules that do not have the liabilities, particularly of the hallucinations. And they've got a very, very strong private company, very strong preclinical valuation justifying large investment. And I think this is where the true pharma play is going to ultimately be for this space going on. It's inspired by those molecules, but this is where I think it's going to move. What we've done is we started off with a compound very similar to psilocybin, which is one of those molecules that we know is active. There's some good data that's recently come out on depression. This is orally active, and we've started making novel, patentable, non-hallucinogenic versions of this drug going forward. And when we actually announced this fairly recently, these are our molecules based on the tryptamine class of psychedelics, and we found that this has got a very fast onset of action. We dosed here animals for about 7 days. They're water soluble, which is very handy when it comes to things like formulation and so forth. And we didn't see any evidence of hallucinations in this case. We were looking for specific behavior that reflected that. And when we tested it head-to-head against Prozac, we saw a very nice response compared to that drug. So, which in this model had very little activity as well. So again, we think we've stumbled on a couple of molecules. We actually have a few of molecules that are similar to this initial one that we based it off. And those were quite promising. So like Delix, which is making large numbers of molecules to test, we're modifying this R group that you can see on this compound here. And we think that our goal is that over the next 6 months, that we will have a candidate that will be ready to advance to the clinic. In this case, very quickly, I won't spend too much time on this as I don't want to leave time for questions and so forth. Our intention is to run another experiment. We're actually looking at molecules that are nothing like the psychedelics to start. So there's very low risk of either abuse or hallucinations. And we're testing some of those head-to-head against ketamine. Our intention is now to build a library of these psilocybin derivatives and then start testing them in vitro and really building a case here for a molecule and get it ready for the clinic, say by Q2. And here we think that we can advance a compound fairly quickly. And again, based on the success that we're seeing with the compound companies like Delix, I think there's going to be a lot of interest going forward with the pharmaceutical companies. And then our timeline is very similar to 204 and how quick we're able to develop these compounds going forward. Very quickly, so our current cap structure and stock information, 32 million shares roughly outstanding, about slightly over 40% insider ownership, I would say now. Reasonable warrant coverage. Cash is starting to come down a little bit since then. Our average burn rate anywhere between 94, 110 roughly. We're looking to get through our toxicology and as I said, develop a second asset with our company with this current round. The executive management here as seen, I'm the president and CSO, and we're getting geared up right now to really advance the company from preclinical as quickly as we can to a clinical-based company and clinical stage company, which hopefully is going to be over the next 9 to 12 months. With that, I'll stop the presentation and I'll open up the floors to any potential questions from viewers.
Excellent. Thank you, Mark. Just a reminder to everybody, you can use the chat function on the bottom of your screen to submit questions. We did receive a few questions in advance as well, so I will lead with those. Our first question asks if, Mark, could you highlight some of the recent commercial transactions of companies in our space and what stage of development they were in, and what do you need at Marvel to achieve to get to a similar point?
Sure, that's a great question. Thank you, Corey. So our goal is to really drive to the clinic, and we want to try and develop a signal in man showing that the drug is active as quickly as possible, because in biotech, typically, historically, the largest single value creation event in terms of percentage change in your company typically occurs when you show efficacy in man. And we see that as, anywhere, say about 12 to 13 months, maybe 15 months out, give or take. And there's two companies that I'd like to highlight, certainly in the neurological space. One's called Athera and the other one's called Anovis. They showed some initial data at the phase 1 type of level that basically saw their valuations reach north of, I think in the case of Athera, depending on when you look at the timing, $500 million, Anovis, $250 million. So we're within striking distance. I can sort of see that. And the interesting thing specifically about Athera is that some of the endpoints that they claimed were relevant, we think we can test in our system as well. So, those are pure pubco plays. And I think those are within our reach in the next 12 to 15 months, I would say, for that case. So there's two examples of pubcos. An example in the NASH space, Arrowhead just did a $120 million deal with GSK with their phase 1 data. So, there's still good interest in the NASH space. That's within striking distance as well for us. And then the real big one is the Delix. As I mentioned, it was a $75 million Series A financing. I think if you look at some Forbes article recently, I think that they are estimating the value of the company at $250 million. They were announced as a Fierce 15 top biotech, which is always a good thing for a company. And so, our second asset, which we parsed the two on, I think that's a reasonable comparable. And, we're in the process of identifying preclinical assets with moving them quickly to the clinic, as is Delix. So, I mean, there's some fairly broad range of valuations that you could consider. And again, Delix, I think you could argue we haven't built up the kind of infrastructure they have, but we do have molecules that appear to be working in our hands too. And we have several of those. And I think in the case of our A2A program, we are, in striking distance of the clinic and potentially increasing our market cap at least an order of magnitude, 10 to 20 fold I think is a potential reasonable target for us to consider at this point. So hopefully that answers the question and gives you a reasonable time frame in which we can accomplish that.
Excellent. Thank you, Mark. Our next question asks, you're obviously a very early stage biotech company, but as president and CSO of the company, what gives you confidence that the compounds that we're working on have a high chance of success?
Oh, that's a great question. So basically, again, just to go back to our drug redevelopment model, I mean, the easiest way is to in some ways don't reinvent the wheel. So in the case of istradefylline, it's the only clinically approved adenosine A2A antagonist that's crossed the finish line. So that's where we started. That drug has a known toxicity profile. It has known pharmacology and so forth. So by starting off with that drug, and again, I go back to the examples of thalidomide and cyclosporin, which followed a similar path. We've made very conservative but reasonable changes by making fluorines, and fluorine occurs in about 30% of all drugs now, roughly speaking. We've been able to make new chemical entities. We think that the toxicology should be acceptable. I don't believe at this point, and nor do I have data that there should be big surprises. So we start off with a known drug, and we're getting it through the process here. And secondly, we're looking at the science. There's an awful lot of science that the target itself is going to play an important role in multiple diseases such as depression, Alzheimer's, and liver disease, and more recently also with oncology plays. So, we think that the asset here is designed to be as safe as we can make it. It looks like it's got a slightly better profile than the original drug, and has multiple shots on net. So I think that's kind of our confidence that we have a good asset here, and our intention is to get in the clinic as quickly as possible now.
Great, thank you, Mark.
Yep.
That's perfect. Shifting gears a little bit here, our next question asks, the psychedelic market is quite active. Why do you think your approach at Marvel is the right approach?
Yeah, again, there's been an awful lot of investment and interest in this space, and good reason to. The antidepressant space has languished for quite a while now. And the idea of neuroplasticity, the idea of rewiring the brain and getting it back up to that can change. That actually has applications beyond depression and anxiety, by the way. The problem is it's potentially, as I like the way Delix put it, it's non-scalable. If you still have to go to a clinic to get treated, or if you have to be under some kind of supervision, I mean, that really curtails the convenience, certainly to the patient. And, some people, I'm sure, will like the idea of the hallucinatory activity. Some people are drawn to that. A lot of people won't be. And the idea of being able to take a pill safely at home will appeal to a lot of people as well. And just reading the tea leaves, at least with the way that the market is turning, I see significant investment going into those patentable molecules. You'll you're now going to get a race to the bottom. In some extent, it's kind of happened to the cannabis space a little bit, as it's been commoditized in some way. So you're going to have to deal with that aspect of it if you're dealing with a known compound and a naturally occurring compound as well. So I like the idea of going after the take it home convenience of molecules that are safe. You have 100%, pretty much as close as you can get to 100% safety in terms of the manufacturing process is very sound because it has to follow FDA, CGMP guidelines and all the regulatory bodies. There'll be excellent proof that these molecules are active. And so patients and medical practitioners can have a lot of comfort that these are safe drugs that they could give their patients. So I see what we're trying to do is the 2.0 version, the next iteration of the psychedelics. And I think that's where it's going. And we're certainly seeing that with some of the companies like Delix in this space as well.
Great, thanks, Mark. Going back to NASH disease, this question asks, why to date have there been no real successful treatments for NASH disease? And what inspires confidence that we have a compelling lead candidate? Maybe you could expand on the recent press release for that question.
Yeah, it's a very tricky question. So I think in our case, I did mention it, and it's worth mentioning again. Our drug istradefylline is actually based off caffeine, and istradefylline, I think, and our molecule follows similarly, is probably anywhere between roughly 100 to maybe 150 times better based on in vitro testing. That's my guesstimate from the literature and our experience with our compounds. And there's some very good epidemiological evidence that coffee consumption can stop things like Alzheimer's, liver-related fibrosis, cancers, and so forth. So epidemiologically, we have reasonable evidence that molecules that go after the adenosine A2A receptor, which is what caffeine, istradefylline, and our MB204 do, could have multiple benefits to these compounds. And so that's powerful. That clinically says that's a viable target. So I think we have that going for us. The second thing is, there's been several groups. In fact, we brought on Dr. Bruce Kronstein, who was one of the original discoverers of the adenosine A2A receptor in terms of liver fibrosis. And since him, other groups have sort of verified and validated the target for liver fibrosis. So, again, there's some scientific rigor there beyond what we've done. There's other groups that have worked with similar compounds or similar, try to go after the same target, and they're seeing similar effects. So, whenever you see multiple groups coming to the same conclusions, that gives you a lot of confidence as well. So, right now, if we do go into the NASH space, as far as I can tell, we'd be at first in class in terms of going after that target. So I think that gives us a fair degree of comfort. And again, istradefylline was an approved molecule to start with. Serotonin, was an experimental HIV drug in its early stages. So regardless of whether or not it's efficacious, we know that the istradefylline is a fairly safe molecule, although I don't believe there's been significant safety signals with Serotonin. So the epidemiology and the fact that other groups have worked on the same target and seen similar effects gives me the confidence that what we've got is good. And again, our drug looks like in many ways has comparable activity to istradefylline on certain things.
Great, thanks, Mark. I see two questions here that go hand in hand, so I will address them. Somebody asks what our monthly burn rate is. That was actually on a prior slide. We're burning about 94 grand per month. Yeah. And with well over $2 million on the balance sheet, that gives us some significant runway here to continue with our efforts. So on that note, we did allocate just about half an hour for this webinar. So Mark, I'd like to thank you for your time, and I will leave it with you if you have any closing remarks here, and then we can finish up.
Yeah, I would say that if there's any people that are investors in Marvel, thank you very much. If there's people considering investing in Marvel, I would say please keep an eye on us. We're trying to hit our milestones as best as we can, and we're moving the company along very quickly. And our goal is, as I mentioned before, is to drive to the clinic to get the massive or as big a value creation event as we can, as quickly as we can. So please keep an eye on us if you're watching the company, and we'll hopefully have more news coming out as we go forward. I think we've got a very good, compelling story. So well, I just appreciate your time for today and listening to the story. Thank you.
Fantastic. Thank you, Mark, and thank you everybody for tuning in today.
Thanks, everyone.